you. And I'm particularly excited to be with you here because, you know, the work is inspired and it's based on the work of the two of the chairmen of this conference, right, which is Yura and Marinka. And hopefully we can actually give justice to their important contributions and the way they actually kind of allowed us to make this work possible. And indeed, I often kind of talk about this project in the context of what did we do for COVID, right? And I'm sure we all had some perspective of like, how did we professionally respond to the COVID emergency? And on our end, when the COVID emergency kind of happened, we realized that many of the network medicine tools we and others in the network community have been actually developing in the last kind of 10, 15 years are really necessary, not only appropriate, but necessary for us to kind of identify new drugs or existing drugs that could cure the disease. So therefore, we ended up putting together a very fantastic team who kind of worked together for about, I would say for about 3 months, very intensively, meeting about 3 times a week, and to really bring these tools to the question how we can really find drugs that may work in the for patients infected with the COVID. Now, to give you a background of how do we actually get started on this process, let's give you a little beginning of how do networks kind of come in when we think about diseases in general and drugs in particular. And at the end of the day, you know, within our cells, there's a very complicated network that consists of multiple layers. and these networks really kind of describe mainly protein-protein binding interactions, but there could be also other regulatory interactions and so on that tell us how the different components, the molecular components of the cell, interact with each other. Now, the question, the scientific challenge, which is as much computational as it is conceptual, is to understand how do we start from these networks and really kind of connect it to human disease. And why there should be a connection is because the breakdown of these components, the breakdown of individual genes, individual proteins, is not a problem by itself. It's often a problem because it affects other components within the network and makes the subnetwork or the machinery within the cell dysfunctional. So the mathematical and the computational challenge is that how do we connect this network to diseases, and eventually how do we use it to kind of think about cures? Now, when it comes to this question, the first question that may emerge, where in this network is a disease? And to kind of give you an indication of that, I'm going to show you very quickly just a network that apparently has nothing to do with the disease, but please suspend disbelief and let's assume that what you see here is a subcellular network in the cell where the intersections corresponds to proteins that interact with each other and the links correspond to the interactions between them. And of course, the reason I'm using this example is because this is a familiar example to everyone across the board. And the question that I would like to really ask is, how is function actually constituted within this network? And because function is not really uniformly distributed, but to be very specific, if you actually want to go to see theater in New York City, you would be most likely going into the Broadway neighborhood because most of the theaters, that is, the entertainment is well located in a particular network neighborhood within New York City. But also, if you're interested in high-level art, like expensive art, then what you would realize that you would go in a different neighborhood. And the way you would actually think about it from the biology perspective is that the network has certain functional modules. And certain functions are well located in that particular network neighborhood. It doesn't mean that some components may not be a little bit far away, but most components who belong to that function interact with each other. They work together to achieve that function. So now, if there are functional neighborhoods and functions are well located in a certain part, then so does the breakdown of a function, which we humans perceive as disease, right? So therefore, we would expect that the components associated with the disease would not be distributed randomly either, but there would be certain neighborhoods that we would call disease modules that agglomerate those components. So for example, you may assume that cancer is somewhere in the vicinity of Wall Street, and you put bipolar disorder around the Times Square, and maybe you can put asthma somewhere next to Washington Bridge, where New York City breeds much of its mess into New Jersey. All right. So in a way, kind of the idea is that these diseases are not just uniformly spread within the network, but they have a well-defined location. If we assume this, if we accept this hypothesis, then the goal of finding a drug or curing a disease is very simple, conceptually, right? You first need to get a map of the city, then find within that the particular disease modules, and then find drugs that hit that particular disease module and not somewhere else. And that's what I mean drugging it. Now, the challenge is, how do you put this one into practice when the network that you have is not complete? And what kind of tools do you actually use to really, get started on this process? And to kind of give you an idea of how this really works, here I'm showing you the protein-protein interaction network of the human cell, the one that I showed you earlier, except now I'm highlighting the genes associated with a particular disease. And in this case, it's asthma. The reason I'm focusing for a second on asthma is because we focused in the lab quite a bit on that disease. And what you can see that most of the asthma genes indeed are located in the same network neighborhood. And some are kind of further away on the sides. And those are far away partly because we may be missing the interactions that would tell us they really belong here, or sometimes they may be mischaracterized as asthma-related genes. So the question is now, how do we take this idea of the disease module now that we have a well-defined neighborhood and transport it to the problem of the COVID, which is not a disease on its own, it's one induced by a pathogen. So to see that, let's kind of go to a more conceptual picture and talk about it. What does it mean to talk in terms of disease module when it comes to COVID-19? Well, the infectious diseases are typically kind of consist of a set of proteins that are injected into the human cell by the virus, right? So in a way, we have actually one network which tells us how the different components of the virus, the proteins within the virus, interact with each other. But when they infect the human cell, so here I'm showing you the human interactome, and this is the viral interactome, the proteins within the virus. And when the proteins of the virus and the RNA that turns into proteins are injected into the human cell, some of the proteins that arrive inside the human cell will actually interact with some of the human proteins. And which means that they will bind to them, and they do so because they need to hijack the mechanism and the machinery of the cell, and they do so by attaching themselves to certain proteins, partly because they want to actually block the cell's defense mechanism, and partly because they want to activate the production of molecules that the virus itself needs to replicate within the human cells. So in that sense, we define the COVID disease module as the set of proteins that directly interact with the viral proteins. So you see, these are kind of part of the module because they're directly interacting with the model. Now the question is, how do we think of drugs? What's the role of the drug? Well, typically the drug has several targets, not only one. And the purpose of the drug is to bind to the human proteins and block certain activities. And what our previous research has shown is that the drugs also form a module, that many of the drug targets happen to be in the same network neighborhood, defined in the drug disease module. So now when we come about the COVID problem, we have a COVID-19 disease module, which is the network neighborhood in the human cell that is directly attacked by the COVID proteins. And then we have the drug disease module, which is the set of proteins to which the drug binds. And the hypothesis is that if the drug binds to the right proteins, it may actually intercept the virus's ability to infect the human cell. If, however, the drug hits in a completely different neighborhood, far from the COVID-19 disease module, then it may be ineffective against the COVID-19. So we need to identify drugs that hit close enough to the COVID-19 module. So the question is, how do we put into this practice? And this research actually started on March 23. And the reason we know that it started there, because that's the date when, thanks to Gordon et al., we actually got the first data that tells us experimentally what human proteins interact with the viral protein. That opened up the possibility to really kind of explore the role of network medicine within the COVID context. And what happened in March 23 is that a starting point, we had actually the human interactome, which is about 18,000 proteins connected with 300,000 protein-protein interactions. And we had the 320 targets of the COVID SARS-CoV virus. And these are 320 human proteins that can bind to one of the viral proteins. And then, of course, we started with a list of about 7,500 drugs, and that altogether had 4,000 drug targets. You may ask, why do we have fewer drug targets than drugs? Well, that's because there are lots of what we call me too drugs. If a successful drug is actually out there on the market, many other drugs pop up who go after the same protein. They try to target the same protein. Now, the question is, what did we do with this starting data? And the first question I said is to ask ourselves, do we have a COVID-19 disease module? That is the virus, when it infects the human cell, is it really hitting a particular network neighborhood, or rather it hits randomly within the cell? And this plot shows you what are the targets of the virus and what are the interactions between them. And you see that we have what we call a large connected component with about 208 proteins. And what we find that this to be statistically significant, indicating that, there's a certain neighborhood where the viral hits or the viral targets agglomerate in the network. So that's what we define to be the COVID-19 disease module. That is the neighborhood where the virus hits. And what we are expecting is that the drug repurposing candidates, because the idea of finding an existing drugs that may also work for COVID-19 is officially called drug repurposing. And we expect that the drug repurposing candidates should also hit somewhere in the neighborhood of this disease module. Now, we should also kind of ask ourselves, is this particular module tissue specific? Because obviously typically the COVID-19 infects the lung. That's kind of his first point of entry. And what we have shown many years ago is that for a disease to manifest itself in a particular tissue is not enough to actually have a disease module in the cell. That disease module has to be expressed in that tissue as well, in this case in lung. So therefore we can use these methods to identify what are the tissues that in principle could be infected by the virus. And what we find is that, yes, it can actually infect the lung. So there's a statistically significant disease module in the lung tissues. But interestingly, and this was a result that we got in March and was very unexpected back then, that we find that multiple brain regions could be affected by that, so that it can be expressed in the brain. And indeed, in the meantime, there's quite a bit of evidence that people infected with the COVID could actually have consequences in the brain and kind of lingers for a while. And we also predicted to be present in multiple reproductive systems, digestive system, as well as cardiovascular tissues. Here, of course, I'm not showing the many tissues for which we predict that it cannot infect, because the proteins that the virus needs are not there, so the disease module cannot show up. Now, the question is, now we have a disease module, so therefore we can start thinking about drug repurposing. And as I mentioned at the beginning, this was not an exercise to apply our tools, but rather what we did is that we took several tools from the literature, some of them developed by our lab, some by other labs, and tested their ability to kind of predict what drugs could work against for COVID-19 patients. And kind of the simplest tool is what proximity-based ranking. And the idea behind that is relatively simple and purely graph theoretical, which is that if these are the viral targets, and these are the targets of the drugs, if they are close to each other, then there is a chance that the drug could disrupt the virus's ability to infect the cell. Now, question is, what does close mean? And for that, we developed a statistical methodology that looks at, is it statistically close to that target, is it close compared to a random selection of drug targets? And that's the criteria that decides whether a particular drug is a potential drug for COVID-19. This is a purely graph theoretical method and that looks really at the path lengths between the drug targets and the wire targets. We also kind of check the diffusion model because diffusion models are widely used for drug prediction. And this was actually implemented by Asher Ameli. And effectively, the question or the philosophy behind the diffusion model is relatively simple. What is looking for uses a diffusion method to identify patterns of attack of the drug target that could really be kind of affecting the virus itself. And then finally, we also applied, thanks to Marinka, and actually she did, a graph theoretical network approach. This is a methodology that she and Yura actually have developed years before. And effectively, the idea is to build a knowledge graph and learn the embedding for every node in the graph. And it turns actually the drug repurposing problem into a link prediction problem, is that to ask, from the learned embeddings, whether a new drug that we're showing to the system could actually have the same effect as some of the existing drugs have for the well-known diseases. And so at the end, what we did is that not only we implemented these methodologies, but because these methods had multiple parameters, we ended up implementing multiple versions of this. So what you see here is that we had 4 different AI pipelines that kind of built and implemented by Marinka, which differed in the way the AI methodology was extracting actually the positive hits. Same in the diffusion, there were 5 different pipelines differing in the parameters and the particular proximity measures used by the diffusion method. And also in the proximity, there were 3 different pipelines that really differed in the terms of the input data. Some of the pipelines use simply the drug targets, others actually use the genes that are differentially expressed if you infect that cell with a particular drug. That was, for example, the P3 line, and P1 and P2 use different simple proximity-based measures. And of course, these methods are not independent of each other and the predictions by them. So we kind of created from each of them a rank list. And that was really, we asked each of them to provide the rank list. And then what we find is that the rank lists within the AI method are very, very correlated. This is the correlation coefficient. So are between the diffusion methods, and so are between the 2 proximity pipelines. The third proximity pipeline is not correlated with the previous ones because it relies on a different data, which is gene expression. So the reason I'm showing this correlation data is to indicate that indeed not all the predictions are fully independent of each other, but there are high correlations within the methods that are not at all unexpected. Now, so once we had this prediction and we asked each method to provide a ranking list from the most likely drug works from COVID to the least likely one, and then the question was, how do we test that? How do we know if we are right? And here we actually got lucky because Boston University has a massive experimental ability, thanks to our level 4 and 5 facility over there, that they were able to work with the virus. And they were anyway planning to kind of do systematic screening of multiple drugs to see which one would work for COVID-19. So we ended up kind of starting to work with them. And what we ended up doing is to kind of develop multiple lines of experimental evidence to see, what is the real positive, what drugs should work and what shouldn't, to test the ability of our pipeline to predict them right. And in particular, we used 2 different classes of outcomes. One of them is based on clinical trials, and one of them based on the experimental testing in monkey cell lines, so-called Vero cell lines. And eventually we follow up with human cell lines as well. I'll tell you in a second. So the first validation is based on the experimental outcomes. And we ended up identifying 918 drugs for which all the pipelines offer predictions and for which drugs were available for experimentation. And why did we do that? Because in principle, the different methods offer predictions for different drugs, and there is some overlap, these 918 drugs, but there's no full overlap between who can predict what based on the data available for that particular drug. So therefore, we wanted to have a pool of drugs for which all methods make predictions, so we can really compare apples with apples and not apples with oranges. So ended up defining this universe of 918 drugs that could be experimentally tested, as well as for which all pipelines have made explicit predictions, so they're comparable. Now, these 918 drugs were experimentally screened for their efficacy against the SARS-CoV virus in vitro 6 cells. These are actually African green monkey cells, and this is a very robust experimental technique, and this is the first line of testing if you want to test a drug in a large scale. And what the experiment showed, that out of the 918 drugs, 37 were what we call strong drugs, which means that they have, at high concentrations, they were particularly efficacious in killing the virus. How the experiment took place, you take a Petri dish of these cell lines, and then you infect it with the virus, and then you add actually the drug, and you do multiple experiments with multiple drug concentrations, and that's what you see here, the drug concentration increasing horizontally. And the question what you're asking is that, will the number of infected cells go down as a result of you adding the drugs? And strong drugs are those that significantly decrease the number of infected cells without killing the cells themselves. Weak drugs, those that have some effect but not as strong as the strong drugs, and there's a criteria there. And there is what we call cytotoxic drugs, about 35 of them in our pool that would simply kill the cells as well. And then 806 drugs of the pool had no effect. The things that we're really excited about are what we call the strong and weak drugs. So 37 plus 40, and these are, we consider, positive outcomes because they show an ability of the virus to kill the drug. And really, whether it's strong or weak, our methodology do not predict the effect size. They just predict that this drug hits in the right neighborhood and could have an effect on the drug. So therefore, for us, a positive effect is a weak effect could also be a positive outcome. And we use negative outcome, the cytotoxic and the no effect drugs, because those are the guys who have no effect over the viral infection. So that's the first kind of experimental outcome. And we also did validation against clinical trials, which means that on April 15, which is the date when we finished the predictions, but we did not yet have the data explicitly, we scanned the clinicaltrials.gov and identified 67 drugs who were in clinical trials at that moment. And we limit that list actually to those that were among the 918 drugs that experimentally tested, and those are 37 drugs. Why did we do that? Because these drugs that were under clinical trials are the best guess of the medical community of what are the potential candidates. So by looking at clinical trials, we are actually kind of capturing the medical community's opinion of what should be the right drug and what should be tested, because if they're testing it in a clinical trial, as you know, that's a very time-consuming and very expensive proposition where lives at stakes. So therefore, you better have a guess of why that should actually work. So now we're going to use these two, experimental and clinical data, to check how well our methodology is working. And so the first thing we measure actually is AUC. And you see that, for example, the proximity does very well in the experiment, but in the clinical trials, the AI pipelines do very well. In other words, AIs are very good to predict the medical consensus. The proximity lines, however, are better to kind of predict the experimental outcomes at the AUC level. But AUC is not a good metric for us. And the reason why this is not a good metric is because AUC really combines the results both at the top of the list and the bottom of the list. In a drug repurposing environment, you just care about the top of the list, right? You want to make sure that there you have the drugs that you care about it, and you don't care so much about the order in the rest of the list. So therefore, precision at 100 is a much better kind of outcome, because it tells us how many of the drugs were really working at the top 100 that my method prioritized. And there you see AI does actually much better. So the second AI pipeline was very good, and so was some of the proximity. And AI, again, was very good at doing precision at 100 in clinical trials as well. And we see a similar pattern when we look at the recall at 100, AI doing very well, proximity doing very well, and so in the clinical trials and in the experiments. But what is interesting about this is that there is no one single pipeline that does consistently well across all the data. So therefore, we cannot just say, okay, A2 is the one and you should always A2, because there are some circumstances where P1 actually does better than A1. So the challenge that came up to us, which was really kind of very interesting for me as a professional advance as well, is that why don't we try to figure out how to combine the outcomes? Because each of these predictions kind of make predictions based on the different principles. And could we get the best of all? Would there be a way to get the best prediction from each of the pipeline? And so therefore, we ended up implementing what we call a multimodal approach, and what you would call a multimodal approach, which is to kind of say, get the best prediction from each pipeline. But the question is, how do we do that? And of course, the simplest is what we call the union list, which is simply say, we're going to trust each pipeline equally, and we're going to take the top K drugs from each pipeline. So if we have 120 drugs to test, we're going to take 10 from each of the 12 pipelines. Of course, we should also check the average rank list, where we just simply look at every drug and its average rank, and then we rank them based on that across all the pipelines. But really what was exciting is to use an alternative, which is to kind of seek what we often call the Kemming consensus, or what your committee calls a Kemming consensus, which is, you know, maximize the number of pairwise agreements between all pipelines. So we don't really care who is first and second and third on the pipeline, but we care what is the relative order between each of them. And we ended up implementing 3 different methodologies to really approximate the Kemming consensus that, as you know, is NP hard to obtain. We implemented Bordascan, Donald, and C-rank, and C-rank is the methodology that Marinka and Jurek have developed previous to this work. And the question is, how do these agglomerations work? And here comes really the punchline, which is kind of the most interesting for us. What we find is that the average rank list offers the worst performance. So here, the higher the line, the better the recall in this case. And you see that the average rank is down here. Actually, that offers the worst performance. Many individual pipelines do better than the average rank. The union list and the downwall do a little bit better, but they're in the middle of the pipeline, right, or predictions. And the best predictions is really often offered by C-rank as well as the Borda. But when we look at across all the different measures, C-rank actually beats all of them, whether we look at clinical trials or whether we look at the experiments. So you see that's kind of the yellow one. And that was really very unexpected to me in the positive sense, right? Is that you can actually have an agglomeration methodology that can really beat all the individual methods. And so this is probably the most important outcome, is that by seeking kind of Kemming consensus through this C-rank algorithm, we can actually combine the best of each of the individual methodologies, and we shouldn't decide who is the best, but rather get the outcomes from them. Now, I should say 2 more things before I finish. The first one is that if you talk with experimentalists, they will tell you it's not enough for you to get predictions from monkey cells, you need to also test them in human cells. But for our purposes, the monkey cells are fine, because what we're really testing here is our ability to predict well within an experimental system. But to complete the paper, we had to do human cells as well. And actually, the human results just came out finally 2 weeks ago. So it's a very recent, and probably Marinka hasn't yet seen that. So this is a surprise for them. And we ended up having, of the top 200 ranked drugs by C-rank, there were 13 positive outcomes in the monkey cells, which include chloroquine and hydrocortisone. We didn't have to test those because those are over-tested. So we tested experimentally 11 of them. And of the 11, we ended up finding 6 that actually worked in the human cells as well. And what is nice is that of the 6, 3 of them are not yet in clinical trials. So these are new predictions that the community was really not fully aware of. And now we're trying to convince actually at the Harvard University clinical trials to take these 3 up, or at least one of them, and see whether they could really improve the disease case of the individuals. And I want to also finish with the second really surprising effect for me, and which is, you know, much of the drug prediction and drug repurposing works on the idea that we are seeking drugs that directly hit the target of the problem, which is in this case, the problem proteins are the direct targets of the virus. And these are what I would call direct hitting drugs. And effectively the hypothesis behind them, if you bind to the protein that the virus needs, then the virus may not be able to bind, and hence it will be effective. What we find that 77 of the experimentally successful drugs, so 76 of the 77 drugs that were experimentally successful are not of this category. That is, that do not hit directly the viral targets. Rather, they are network drugs, that they induce network-based perturbations that alter the virus' ability to enter the cell or replicate with it, without hitting directly in the immediate kind of interaction partners of the drugs. And why am I excited about this? Because it shows that the traditional methods that all rely on binding will not be able actually to identify these type of drugs, and really network methods, network science-based methods are the only way to find them. So this really opens up not only for drug repurposing, but also for drug development, new avenues that are indirect acting drugs that could be effective. And I should finally just briefly mention that there are many other efforts out there, and probably many of them listing, that are trying to develop drug repurposing methods with great success for COVID. And, you know, we should kind of look at them as a common effort by the community. And finally, I will end up by thanking the fantastic team who made that possible, both the computational and bioinformatics team who is shown here on the plot on the top, as well as the experimental team that has kind of heroically worked on this very severe environment to really test the predictions. And I'm delighted to kind of stop here. I should mention, as the previous speaker did, we are also looking for talented postdocs, not only always, but right now in the hiring phase. And we have lots of cool projects in the area of network medicine and AI. And if you would like to kind of join us, so we're delighted to see your application. Thank you.
Thank you very much, Laszlo, for excellent talk. Always super exciting and interesting. We have a number of questions, and we have about 4 minutes to answer them. So the first question is about the disease module concept is quite interesting and pretty much the foundation on which a lot of methodology is built. But what about functional crosstalk between pathways and or biochemical processes? What would be your response to this question?
Sure. And one, of course, the traditional way is to identify the disease pathways and then kind of look at targets that attack those. And we do that a lot, but we find actually that most diseases are really talking to multiple pathways, and there are kind of a crosstalk between the multiple pathways, and certain part of the pathways involve the disease and other parts are not. So the disease module is not independent of the pathway. It includes multiple pathways, but it kind of offers a much more well-localized, network sense, well-localized neighborhood of the cell. So the pathways are longer. On the other hand, what we often do is that once we find a disease module, to understand what is the role of that network neighborhood, we look for enrichments in particular pathways within that, and that tells us what functions are being collected within that disease module.
A follow-up, thank you for this would be, if I understand the question correctly, is how are you thinking about the difference between associations versus some kind of causal or consequential type of links modeling, let's say some processes and things like that? How do you reconcile these two views?
Yes, this is a fabulous question. And here I'm looking at your community to help us with that. This is obviously a major problem in the case of network medicine, partly because the experimental tools are still in the early stage, as well as partly because you know, because the tools are just being developed by your community. On the other hand, one of the reasons we actually designed the experiments we did is to have a blind set, and we can able kind of say that at least the methodologies are predictive, and we're hopefully kind of dividing us causal relationship that we have in the cell. But I think this is a journey that we entered together with you, say about 5-10 years ago. And I think we're really looking at your community to help us understand that we mean more like the CS community.
Thank you, Laszlo. And then a couple of questions I'll kind of summarize and paraphrase is about whether the interactome can be dynamic, or whether it can change over time, and whether it could be, let's say, phenotype specific. Can you comment on this?
Yes, so certainly the interactome as a whole is just what I would call the architecture of the network, and only subsets of that network are active in any environment. The first restriction we always do is to look at the tissues dependence if we have a particular tissue where the disease is kind of manifested. But obviously there's lots of dynamical aspect of when the certain proteins are present or not, even within the cell cycle. Ideally, we would like to build into those tools into our prediction system, but unfortunately, there's not enough dynamical information to be able to do that. This is where I often think that the success of network medicine and network science is all relies on static data. And the reason why we don't have dynamical approaches is not because we don't think dynamics is important. We think dynamics is hugely important, but the experimental tools are not there to provide dynamically resolved information at the molecular level at the large scale. There has been attempts with microarray tools and things like that to get at least snapshots, but those are not at a single cell level, but at the large colony level, millions of cells together. So this is, I often think that both within network medicine as well in network science, one of the big puzzles for us is dynamics at large scale, and to develop the tools that can really take advantage of the dynamical information when it comes.
Super, thank you very much.